So if you have single-end data, and in RNA-seq you will have multiple samples, and you will typically have two groups of samples. That's why we have the white files and we have the black files. So these are two control samples, and these are two treated samples. Yeah? In reality, I advise you to go for at least 4 in each group, but then this drawing would be too complicated. So I take 2 here, but in reality, go for at least 4 in each group, then you're statistically safe. Okay, so how does it work? Well, when you have single-end data, then each sample is one FASTQ file. In FastQC, you're going to process each FastQ file individually. So you get 4 FastQC reports. You're going to clean your FastQ files with Tremomatic, and again, you're going to do that on each file individually. You can do it in batch, but it will be in each file individually, so the output will still be 4 FastQ files, 1 for each sample. Then you're going to map, and again, you're going to map each file individually. Yeah? Going to map this and this and this. And if you do a 2-pass, he's going to take information, splice junctions from all the mappings, from all the files for the 2-pass mode, but still he will map each file individually. So after mapping, you will get 4 BAM files, 1 for each sample. Then you will typically do the counting. Still, it depends there, but some counting tools will still process each file individually. Yeah, like HT-seq count, the one that we typically use in the RNA-seq course. He will generate 4 counts files, 1 for each sample individually. Now there are counters that produce 1 output. But even these counters, like feature counts, that's another one, even these counters will process every BAM file individually and will create initially 1 count file for each sample, but they will then combine these 4 count files into 1 matrix. Yeah? And it's only here typically when you go to R, so this is what you do in Galaxy, and then you go to R to find differentially expressed genes. And it's only there that the individual counts, so for each sample individually, are combined into 1 matrix, where each sample becomes 1 column. And this is what you analyze. But even here, each sample stays individually a separate column in the matrix. There's no step where you're going to combine all the counts from these biological replicates. Yeah? You need them individually for doing statistics. If you're going to combine them at some point, you lose your statistical power, so you don't do that. So this is how it works. For paired end data, the workflow is a bit more complex, because you will start with 8 files. Yeah? 2 FastQ files per sample because it's paired end. So in FastQC, you're going to process them individually. So for each individual FastQ file, you will generate a report. So even the 2 FastQ files from the same sample will not be taken together. So 1 report for each FastQ file here. Tremomatic will use information from each pair of FastQ files, but the output will still be 1 FastQ file per FastQ file here. So still 8 files in total. Yeah? He will take them together because he's going to use the information from the 2 files to find adapter contamination, but the output will still be separate. And it's only here during the mapping that he's really going to combine the 2 FastQ files from 1 sample into 1 BAM file. So he's going to map them together and will produce 1 BAM file for the whole thing. And then the rest of the workflow is the same as here. So you have a counting tool that produces 1 count file per sample, and then they are combined in R when you do the ESEQ2. They are combined into 1 matrix where every sample is one column.